There is a population living on this planet that no geneticist in any laboratory with any tool can fully map. Not because the science is not advanced enough, not because the data is missing. It is because this population carries more genetic complexity inside their chromosomes than any other group of human beings on Earth. A combination so rare and so historically unprecedented that researchers at the Broad Institute of MIT and Harvard describe their genomes as a mosaic unlike anything else in the human species. That population is Black Americans. And the reason why stretches back not just 400 years to the slave trade, but 300,000 years to the origin of the human species itself. It connects the oldest DNA on the planet to one of the most painful chapters in modern history. Before we dive in, comment where you are watching from and click subscribe. Let's establish the foundation first, because to understand why Black Americans are genetically extraordinary, you first have to understand why Africa itself is genetically extraordinary. Modern humans, Homo sapiens, evolved in Africa approximately 300,000 years ago. For the vast majority of our species' existence, all of us were African. Every human lineage on this earth, no matter how far it has since traveled, traces back to that single origin point. And then somewhere between 60,000 and 70,000 years ago, a group of humans left. Not the whole population, not even a large fraction. A relatively small group departed Africa, crossed into the Levant, and began the slow colonization of the rest of the world. Europe, Asia, the Americas, the Pacific. Over tens of thousands of years, those small founding groups spread, adapted, and diversified into every non-African population on Earth today. Here is where the science gets important. When that group left Africa, they took something with them, and they left something behind. What they took was a small sample of African genetic diversity. What they left behind was everything else. Think of it like drawing a cup of water from the ocean. The cup contains water, real water, but it is a tiny fraction of everything the ocean holds. The cup carries some of the diversity of the sea. The ocean carries all of it. Africa is the ocean. Every other population on Earth is the cup. And as those founding groups spread further and further from Africa into Central Asia, into Europe, into East Asia, and eventually across the land bridge into the Americas, each successive migration repeated the same pruning process. A small group broke off, took a subset of the diversity available, left the rest behind. Geneticists call this a serial founder effect. Each step away from Africa is another cup drawn from a progressively smaller vessel. By the time you reach the indigenous populations of the Americas, you are looking at a genetic library that, though still rich and complex in its own right, represents a thin slice of what began in Africa hundreds of thousands of years earlier. The consequence of that serial pruning is visible in the genome to this day. Non-African populations share longer stretches of identical DNA with each other because they descend from those small founding groups. The variation that was lost at each bottleneck simply no longer exists outside Africa. It stayed behind. It stayed in the continent where humans were born. This is what geneticists call the founder effect, and the numbers it produces are staggering. According to research published across multiple institutions, including the University of Pennsylvania, the Wellcome Trust, and the Human Genome Diversity Project, African populations harbor more genetic diversity than all other human populations on Earth combined. Not more than Europe, not more than Asia, more than Europe plus Asia plus the Americas plus the Pacific, all added together. The average nucleotide diversity, meaning the rate at which DNA sequences differ between two randomly chosen people, is nearly twice as high in African populations as in European or Asian populations. Twice. That gap is not a small statistical noise. It is a fundamental signature of deep evolutionary time. Sarah Tishkoff, a geneticist at the University of Pennsylvania who led one of the largest studies of African genetic diversity ever conducted, spent over a decade trekking across Africa collecting DNA from 121 distinct African populations across 13 countries. What her team found, published in the journal Science, confirmed what the theory predicted, but made it viscerally real. The genetic distance between two neighboring African ethnic groups, for example, the Yoruba and the Igbo, who live within the same region of Nigeria, can be greater than the genetic distance between a European and an East Asian. Let that land for a moment. Two groups from the same corner of one African country can be more genetically distinct from each other than a Norwegian is from a Japanese person. And yet both of those groups, and dozens more like them, are simultaneously represented inside the genome of a single Black American whose ancestors were enslaved 4 centuries ago. That is the depth of diversity that exists within Africa. That is what 300,000 years of human evolution looks like when you have not been pruned by a founder bottleneck. Now here is where Black Americans enter the picture, and here is where the story becomes something no other population on Earth can claim. Black Americans did not descend from one African ethnic group. They did not descend from 2 or 5 or 10. The transatlantic slave trade, one of the largest forced migrations in human history, drew enslaved people from dozens of distinct ethnic groups across the entire western coast of Africa, stretching from Senegambia in the north all the way down to the Congo-Angola region in the south. Researchers at the University of Pennsylvania and Cornell analyzed 300,000 genetic markers from across the genome and described the result with a single phrase that has since been repeated in study after study. They called it the rich mosaic of African American ancestry, a mosaic, not a single tile, not a simple pattern, a mosaic, hundreds of distinct ancestral pieces assembled by violence and history into something that has no parallel in the genetic record. The Yoruba, the Igbo, the Wolof, the Mandinka, the Akan, the Bakongo, the Fula, the Isan, the Senegambian groups, the Windward Coast peoples. Study after study from Stanford, from MIT, from Penn, from Oxford finds that the African component alone in a typical Black American genome draws from multiple genetically distinct West and Central African populations simultaneously, not sequentially, not as distant ancestry that has been diluted over millennia, but as active, traceable segments of DNA woven together within living people over the last 10 to 15 generations. And that is only the African component, because Black Americans did not stop there. A 2014 landmark study in the American Journal of Human Genetics, using genetic data from hundreds of thousands of individuals through 23andMe, found that the average Black American genome is approximately 73 to 75% African in origin, roughly 24% European, and about 0.8 to 1.2% Native American. 3 continents in a single person's DNA. No other major population on Earth carries this specific combination. Not Latinos, whose admixture is primarily Iberian European plus indigenous American. Not the mixed populations of the Caribbean, who carry African and European ancestry, but with different regional sources and different proportions. Not any population in Europe or Asia. The specific 3-way combination of West and Central African, Northwestern European, and Native American ancestry, woven together in the particular proportions that characterize Black Americans, is found nowhere else on the planet in this form. And each of those 3 components carries its own deep genetic complexity. The African component alone draws from dozens of ethnically and genetically distinct populations. The European component is predominantly British and Irish, as confirmed by Oxford University haplotype analysis, but with smaller detectable contributions from French ancestry in communities with ties to Louisiana and other historically French colonial territories. The Native American component, while smaller, averaging around 0.8 to 1.2%, reflects centuries of direct contact between enslaved Africans and indigenous peoples on American soil, contact that the written historical record barely documented, but that the genome preserved with perfect fidelity. When you layer all three continental sources together, each one itself a composite of multiple distinct populations, you arrive at a genome of extraordinary combinatorial depth. Researchers at Dartmouth described it as one of the most complex genomic structures in the study of human populations. Not the most complex in one dimension, the most complex across multiple dimensions simultaneously. If you are new to this channel, subscribe now. We build every video from peer-reviewed science and go places mainstream history never goes. What comes next is the part most people have never heard. Here's what makes the Black American genome not just diverse, but scientifically extraordinary, in a way that researchers are only beginning to fully understand. When geneticists study a population to find the genetic causes of a disease, they rely on a property of DNA called linkage disequilibrium. This refers to how tightly genetic variants cluster together along a chromosome. In populations that went through a founder bottleneck, like Europeans, large blocks of DNA travel together as a unit across generations. Those long blocks make it hard to pinpoint exactly which gene within the block is causing a particular trait or disease. Researchers can narrow things down to a neighborhood, but not to a single house. African populations, because they never experienced that bottleneck, have dramatically shorter haplotype blocks. Their DNA recombines more frequently. The genetic neighborhoods are smaller. This means that when researchers study African ancestry populations, they can pinpoint a disease-causing gene with far greater precision. David Reich at the Broad Institute of MIT in Harvard, one of the world's leading population geneticists, led a study published in Nature that created the most detailed genetic map of the African American genome ever assembled. His team analyzed the DNA of 30,000 Black Americans and found approximately 70 recombination events per person's genome. They also identified a novel 17-base DNA sequence found in people of West African ancestry that does not appear in any non-African population studied, a sequence that exists nowhere else in the human genome record. The implication is staggering. There are variants in Black American DNA inherited from West African ancestors that may hold the key to understanding diseases that affect all of humanity, but that have never been identified because the vast majority of genetic research has been conducted on European ancestry populations. The New England Journal of Medicine published a commentary on exactly this point, noting that African populations have the greatest genetic diversity of any group on Earth, and that this diversity makes them uniquely powerful for biomedical discovery, not as a subject of study in some extractive sense, but as carriers of the full range of human genetic information, the complete archive of what our species is capable of. And Black Americans sit at a unique intersection within that archive. They carry the ancient depth of African genetic diversity, the 300,000-year-old well of variation, combined with European and Native American ancestry that arrived within the last 10 to 15 generations. The result is a genome that recombines faster, encodes more variation per chromosome segment, and contains genetic information that exists nowhere else on the planet in quite this combination. Researchers at Frontiers in Genetics noted that genomic studies in Africa have received less than 1% of total global investment in genetic research, less than 1%, for the population that holds the deepest and most complete archive of human genetic history. And yet, scientists are increasingly recognizing that unlocking the full map of human genetic variation for disease research, for drug development, for understanding evolution itself requires looking here, not less. Now let's talk about what this means at the individual level, because when geneticists at Dartmouth, Vanderbilt, and McGill published a comprehensive study of African American genomic diversity in the journal PLOS Genetics, they described something that should stop anyone in their tracks. They found that individual Black Americans carry DNA segments that can be traced simultaneously to West African populations, to specific European ancestry clusters, and in some cases to Native American lineages. and these segments sit side by side on the same chromosomes, in the same cells, in the same living person, not as ancient echoes that have been smoothed out over thousands of years of intermixing. They are identifiable, traceable, geographically mappable pieces of ancestry, each one belonging to a distinct historical thread. Imagine a library where every book comes from a completely different civilization: ancient West African kingdoms, colonial British settlers, indigenous North American nations, each book written in a different language, carrying a different history, encoding a different way of surviving the world. And then imagine all of those books bound together into a single volume that is simultaneously all of them at once. That is the Black American genome. There is one more dimension to this that the science has documented, and it is the one that tends to silence rooms. The genetic uniqueness of Black Americans was not the product of choice. It was not the result of exploration, trade, or cultural exchange. It was the product of the most violent, forced migration in recorded human history. The transatlantic slave trade did not move people across the ocean in any single wave or from any single source. It drew from dozens of African kingdoms and ethnic groups, mixed them together on plantations across the American South, and then subjected them to centuries of further forced intermixture through slavery's systematic sexual violence against enslaved women. Every layer of complexity in the Black American genome has a name attached to it. The Yoruba thread is the memory of the Kingdom of Oyo. The Bakongo thread is the memory of the Kingdom of Congo. The British-European thread is the memory of men who called themselves owners. The Native American thread is the memory of forced coexistence on land that belonged to neither group. The genome is not neutral, it is a ledger. And the ledger of Black American DNA is the most complex human accounting in existence. What makes this even more remarkable is that the genome encoded all of this involuntarily. Black Americans did not curate their ancestry. They did not choose which lineages to carry. The slave trade chose for them. The plantation system chose for them. The domestic trade that moved over 1 million enslaved people across state lines after 1808 chose for them. The systematic rape of enslaved women by their enslavers chose for them. Every violent rupture that was supposed to erase identity instead left a biological signature. Every forced mixing that was intended to sever people from their origins instead created a new and extraordinary genetic record. The archive that slaveholders tried to destroy is permanently written into the chromosomes of their victims' descendants. It cannot be burned. It cannot be redacted. It cannot be legislated out of existence. It persists, intact, in tens of millions of living people. And when geneticists study that archive, when they sit with the raw data, the principal component analyses, the admixture plots, the haplotype maps, they find not just complexity, they find a genome that carries more human history per chromosome segment than any other population that has ever been studied. In 2009, Sarah Tishkoff and Carlos Bustamante published their landmark study in the Proceedings of the National Academy of Sciences. It analyzed 300,000 genetic markers across West African and African American populations. Among their conclusions, stated plainly in the abstract, was this: the genomic diversity among African and African American populations is far more complex than originally thought. Far more complex than originally thought, from two of the most respected population geneticists in the world. What they meant, what the data showed, is that the tools scientists had been using to understand human genetic diversity were not built for what they were looking at. The variation was too deep, the combinations too layered, the mosaic too intricate. They were looking at the oldest DNA on Earth, refracted through one of the most turbulent human events in history, expressed in a living population of tens of millions of people, and it had no equal. No other population on Earth sits at this particular genetic crossroads. European populations are genetically younger, their diversity shaped by the bottleneck of the out-of-Africa migration and subsequent isolation. East Asian populations carry the deepest ancestry from that eastern branch of the out-of-Africa dispersal, refined by tens of thousands of years of geographic separation. Indigenous populations of the Americas carry a separate and ancient lineage, shaped by the migration across the Bering Land Bridge. Each of these populations is remarkable in its own right, but none of them carries what Black Americans carry: the full depth of the oldest human genetic lineage on Earth, plus the imprint of every violent continental collision of the last 400 years, expressed in a single living genome. Scientists used to think genetic uniqueness was about isolation, about populations that stayed in one place long enough to develop distinctive traits. The San people of Southern Africa, the most ancient human lineage on Earth, represent that kind of uniqueness. Ancient, deep, unmixed. What Black Americans represent is something else, something the science had no category for until researchers sat down and actually mapped it. Not the uniqueness of isolation, the uniqueness of convergence, the uniqueness of a genome that contains everything, because history gave them no other choice. That is what the science found. Not a story of diminishment, not a story of mixture as loss, a story of a population that absorbed the genetic weight of 3 continents, survived what should have been unsurvivable, and arrived in the present carrying the most layered, most complex, most historically saturated genome on the planet, the oldest lineage, the deepest diversity, the most continental threads, the most recombination events per chromosome, the genetic variants that exist nowhere else. The science has a word for all of that: extraordinary. And it is a word that the data, not sentiment, not politics, not cultural pride, but the raw, peer-reviewed, replicable data earns completely. If this video opens something for you, share and subscribe.